Okay, hi everyone. My name is Dave Julian, and we are from K1G8. And today we are going to present about nucleic acid. So these are my group members, which is Mohammad Najib, Lina, Dira, and also Eng Xuexuan. Okay, so before we go into the deeper parts of nucleic acid, so let's just talk about simply a basic. What is nucleic acid? So actually, nucleic acid is an important class of macromolecules found in all the cells and also viruses. And the function of nucleic acid have to do with the storage and expression of the genetic information. So as we call it DNA, the deoxyribonucleic acid encodes the information that the cell needs to make protein. So a related type of nucleic acid called ribonucleic acid, which called RNA, comes in different molecular forms that participate in the protein synthesis. So this is nucleic acid. So for my part, I'll be presenting about the structure of nucleic acid. So the nucleic acid is a polynucleotides, which are long chain-like molecule composed of a series of a nearly identical building blocks called the nucleotides. So what is the nucleotides? As we can see here, all is the nucleotides, which in RNA, the nucleotides we found is cytosine, guanine, adenine, and also uracil. And for DNA, it will be different in this one. So the uracil will be changed in the timing. Means the first three, which is cytosine, guanine, adenine, will be the same, but the uracil will be changed in the timing in DNA. Okay, so each nucleotide consists of a nitrogen-containing aromatic base attached to a pentose sugar, which is in turn attached to a phosphate group. As we can see here, the we know that all the aromatic base is some kind of like benzene or the 6 but for benzene it will be 6 part of carbons in all the blocks, but in this one the nitrogen base will be have nitrogen here, and this part is the nitrogen base which is connected to the pentose sugar here with the pentagon ties, and also lastly connect to a phosphate group. Okay, so for simple things that we know that in DNA and also RNA, the different types is the pentose sugar. They'll be different in the DNA, which is the 2' deoxyribose. It's different from the sugar in RNA in the ribose by the absence of hydroxyl group on the 2' carbon of the sugar ring. As you can see here, the 2' carbon sugar ring. Both of these is the same pentose sugar, but in deoxyribose, it will be consist of hydrogen here, but in the RNA, which is the ribose sugar, will be consist of hydroxyl group in here. So that ends my part. Thank you.
Thank you to the previous presenter. Next, I'm going to explain about the functions of nucleic acids. So, nucleic acids serve as a primary information carrying molecules in cells and make up the genetic materials. It's also function to transmit and express that information inside and outside the cell nucleus to the interior operations of cells. It also play important role in directing protein synthesis. And also, nucleic acid has a specific roles in sequencing of DNA. For example, nucleobase pairs enable storing and transmitting coded instructions as genes, while in RNA-based pair sequencing provides for manufacturing new proteins that determine the frame and parts, and also most chemical process of all forms. And that's all for my point. Moving on to the next presenter. Thank you.
Good day, everyone. My name is Diraki. The metric number is 210638. I will be talking about purine and pyrimidine. Okay, so first of all, both of these bases, which are purine and pyrimidine, are a heterocyclic aromatic organic compound. But what distinguishes them is the structure. As you can see here, okay, in purine is a double ring with the pyrimidine ring is being fused to the imidazole ring here. And pyrimidine is only a single ring. with having two nitrogens at position 1 and 3. And okay, so the molecular formula for the purine is C5H4N4, whereas for pyrimidine it is C4H4N2. Okay, next slide. Next slide, I will be talking about the two purine bases and also the three purine bases. So we know for the purine structure, it consists of a double ring, unlike the pyrimidine, which only consists of a single ring. Okay. So for the two purine bases, we have guanine and also adenine. There are the other name for each of them. So for guanine, it is also known as 2-amino-6-hydroxypurine with the molecular formula of C5H5O and 5. As for adenine, it is known as 6-aminopurine with the molecular formula of C5H5 and 5. Next, moving on to the pyrimidine bases. First is uracil, which is known as 2,4-dihydroxypyrimidine with the molecular formula of C4H4O2 and 2. Okay, the second pyrimidine bases are thymine, which is known as 2,4-dihydroxy-5,5-methylpyrimidine with the molecular formula of C5H6O2 and 2. Last but not least is cytosine, which is known as 2-oxo-4-aminohydropyrimidine with the molecular formula of C4H5O and 3. Next slide. So the current slide is showing about the nucleic acid and the nucleotides. So first of all, I will introduce what is meant by nucleoside and what is meant by nucleotides. So nucleoside is a compound formed by the union of the nitrogen base and also a pentose sugar. Whereas for the nucleotides, it is a compound formed by the union of a nitrogen base, a pentose sugar, and also a phosphate. Okay, so as you can see here, the ring of the position of the sugar component of the nucleosides, okay, is indicated by the prime number. For example, 5' and 2'. So the nucleosides of the RNA is precisely known as ribonucleoside. Whereas for the nucleosides of DNA, it is known as deoxyribonucleosides. So at here, by referring to the table here, ribonucleoside is the RNA and deoxyribonucleoside refer to DNA. How to remember is R is belong to RNA and D is belong to DNA. Yeah. So in RNA, okay, I repeat again, the 5-member ring sugar is D-ribose. And in DNA, the 5-member ring sugar is 2' deoxy-D-ribose. It is the D-ribose without an OH group in the 2' position. Okay, by referring to the table here, each of them have their own names for the ribonucleosides, deoxyribonucleosides, ribonucleotide, and deoxyribonucleotide. I will focus on thymine and uracil here. Okay, so you must be wondering why thymine doesn't have they have no names in the ribonucleoside parts, and why uracil doesn't have their own names in the oxyribonucleoside parts. So let me explain. Because uracil it is only discovered in RNA here, it is attached to the ribose, but not to the deoxyribose, which present in the oxyribonucleoside here. In thymine, it is found only in DNA. That's why in the DNA it is known as thymidine, but not in ribonucleoside because it only attached to 2-deoxy ribose, which only be discovered in DNA, but not in the ribose in RNA. So in the nucleotides here, you can see that for adenosine is known as 5' monophosphate, adenosine known as 5' diphosphate, and for the other adenosine it can also be known as 5' triphosphate. In nucleotides, it is easy to distinguish them because at the name of the nucleoside you can add mono, di, and also tri. So this, as you can see, the mono here is only have one chain. In diphosphate here, it has two chain of the same structure, and for the triphosphate, it has 3 repeated chain, 1, 2, and 3. So it is named as triphosphate. Okay, so the same goes to the nucleotides of 2' deoxyadenosine here. One chain known as monophosphate, 2 chain got diphosphate, and 3 repeated chain here known as triphosphate. So that is all from me. Thank you.
Hi everyone, I am Lina Turkestani and I will be talking about deoxyribose nucleic acid, aka DNA. DNA carries genetic information in all living organisms. It consists of 2 strands that take the shape of a double helix due to the strands wrapping around one another. The DNA structure is in an anti-parallel arrangement. This means that the first strand runs from the 5' to the 3' and the second strand runs from the 3' to the 5'. DNA has phosphate groups and alternating sugars that make up the backbones of the structure. Then the inner structure are the 4 bases: adenine, thymine, cytosine, and guanine. In this picture here, you can see the anti-parallel arrangement and the double helix structure of DNA. The DNA base pairs, adenine will always connect to thymine and guanine will always connect to cytosine. The bonds that hold DNA together are the phosphodiester bonds and hydrogen bonds. The phosphodiester bonds are covalent bonds that are the linkage between the nucleotides and the phosphate groups. The hydrogen bonds are found between the bases, when A is attached to T and C is attached to G. In this photo here, you can see the phosphodiester bonds, as well as the hydrogen bonds in between each of the bases. There are 2 hydrogen bonds between T and A, and 3 hydrogen bonds between G and C. These are the different 3D forms of DNA.
Okay, now I'll talk about ribonucleic acid, which is RNA. It consists of single polynucleotide, which is composed of a ribose sugar, phosphate, and one of the 4 nitrogenous bases, which is adenine, uracil, guanine, and cytosine. Uracil is found in mRNA instead of thymine. There are 3 types of RNA, which is messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA contains complementary codon formed by the transcription of cisdron of a single DNA strand. Carries the genetic code, which is transcribed from DNA in the cell. They make up about 3 to 5% of the total RNA in the cells. Transfer RNA is the smallest RNA molecules that are folded into 2D clover leaf shape, held by hydrogen bonds, which will then folded to become 3D L shape. The 3 N always end in the base sequence of CCA and act as the amino acid by this side. Its anticodon is always complementary with the codon in the mRNA and is directly related to the type of amino acid carried by it. Different tRNAs carry different amino acids to the ribosome for polymerization into polypeptide chain. They make up about 50% of the RNA in the cells. ribosomal RNA is synthesized in the nucleus. Thus, the aggregates contain in the nucleus to form the large ribosomal unit and small ribosomal unit. The subunits which is synthesized in the nucleus leave the nucleus by nuclear form and combine to form complete ribosomes. They make out about 80% of the cells.
Okay, so that's end our brief discussion about nucleic acid. So thank you for hearing and watching our video. We are from K1G8, and I hope you guys enjoy it. Thank you.